Put your mask on and I'll. Alright, does it feel familiar? Swirl it around. So, let's have a think about it. They're both the same. What's different? Maybe it's how they're prepared, maybe it's the brand. Just give it a think for a few seconds.
Finally, okay.
And like I think maybe it's more like a friendship thing with us.
I'm gutted right now.
Oh, Chaz, that was just unpheasant.
Feel like I've been strung along. Very unhappy bird right now.
I'm a little relieved at this point because it's not awkward. I think he's taking it as well as anybody would.
I'm not sure that's his taking it well face. Anyone for a dance?
Please, could everyone gather in the cabana immediately?
What?
Give me a break, Lana.
Call me psychic, but I think we're about to see a group of grown adults dressed as giant birds get pulled off by a talking comb.
Lana just jumps in. The mood is flipped upside down. She knows how to ruin everyone's party.
I have no idea why she called us here. I've been good.
Well, Miss Loophole, you ain't above the law.
I have gathered you all to remind you that this retreat is for finding deeper connections, not just physical ones. This information is not getting through.
and it means that there's a lot of protein in that particular sample. So it helps you to quantify in reference to a known amount. So if you have, say, a microgram of a particular protein and you load it, and that band gives you a certain size, and then you have another band which is the same, you can say, well, actually, I've got approximately 1 microgram of protein. And if you know the size of that protein that you've loaded, it's 50 kilodaltons, and your protein is running at the same size, you can again say, well, it is approximately 50 kilodaltons in size. So it allows you to have those approaches to actually quantify and get some physical characteristics of a protein. And remember, this is about a protein that perhaps you're purifying, because you don't know anything about it. You've identified a novel protein. It's something that's expressed during a particular stage of cancer. It's something that's expressed following a treatment with a drug. And this is the protein that you're interested in, because when you treat that person with that drug makes this protein which gives those beneficial effects. So there's all these things that you may not know about the protein. Okay, excellent, thank you. And so the last one then is the basic idea regarding our two-dimensional STS page. And I'll just preempt, remember you start off with a one-dimensional and then you move on to a two-dimensional approach. So first of all, why? And then what is the process? What are the characteristics that we are now going to use? Because they must be different to the one-dimensional, because we're looking at the migration in a different context. Haley first focuses on separating proteins by isoelectric focusing. Good. So my question would be, well, what does isoelectric mean? Or what is the isoelectric point of a protein? So everyone else can jump in too.
A certain pH at which the net charge of the protein is zero.
Very good, thank you. So isoelectric point is where you have the net charge. The net charge is the overall charge. So it doesn't mean that there's no charge on the protein. It means that the number of positive charges is equal to the number of negative charges. So when you then, based on what we've just said with the one-dimensional, that when you have a protein with some charge and you apply a positive or a negative current, that protein will move. So if the protein is positive, it goes to the negative. If it's negative, it will move to the positive electrode. So when there is no net charge, it stops moving. Now, if we just focus and keep going in terms of the isoelectric charge, how do we change that charge on the protein? What do we need to change to the media? What characteristics of the media do we need to change to affect that charge of the protein? pH, perfect, thanks Haley. So we have different pHs now. And thanks Aaron, you said the same thing, which is great. So pH, there are a group of compounds called ampholytes. And basically what this is that you have a gel. Within that gel you have a matrix. And within that matrix you have one end which has one pH, and the other end you have a different pH. And you can go from 0 to 14, or you can go from 0 to 3, or you can go from 7 to 14, or 9 to 14. You can go from an acid, neutral, or a basic region. And when you put your protein in that sample, you've already separated them based on size, which is the one-dimensional. Putting them into the two-dimensional then says, okay, these are all the different sizes, but they might have different charges. And so when you apply that at that particular pH, any protein that has a different isoelectric point is going to move either to the positive or to the negative electrode. And once they reach that point, they then stop moving. Because it's possible that you can have two proteins or more that are exactly the same size, but they're different proteins. Okay? And because of that, if you just do a one-dimensional gel, you might see a single band and think, oh, there's only one protein there. But because they're running at the same point, you can't differentiate between the two. So what you have to do in this context is run the two-dimensional gel. And so that then says, okay, they're all running at the same point, but then they start to migrate apart because this protein has a different isoelectric point to this one. That tells you, actually, it's not 100% pure, you've got 2 units. Now, if you do that and you only see one band, you can then be much more confident to say, I've only seen one band that runs at 50 kilodaltons. In the isoelectric point, there's only one band and it's shifted across at this point, and its isoelectric point is 8.2. You know that it has a net negative or net basic character because it's only migrated to that location. So it gives you additional information. So as I said, follows 1D, you separate them based on pH and is used to confirm purity of that particular mixture. So that's really quite an important way of determining purity of that particular protein and that construct. Okay, are there any other questions? So feel free to unmute or put it into the chat relating to this particular lecture. Or just let me know on the screen with an emoji or something how you're feeling about this lecture. Thanks, Amy. Again, sorry, I'm probably mispronouncing your name. Everyone else? Myra, Chelsea, thank you. Okay, it seems like everyone's happy, which is good. And remember, these tutorials, if you've got content from last week's tutorial, please don't think, oh, I can't ask a question about last week. You can ask questions about the content at any time, and I really encourage you to ask questions about the content throughout the semester. I'm starting to get students emailing me about different questions about enzyme kinetics, etc., which is fantastic. We heard a question right at the beginning about the practical. That is fantastic. Keep it up, okay? Do that, engage with the content. Even if it's a small question, doesn't matter. It's better than no question when you have one, so please keep asking. Okay, so I'll stop that one. Let me just change my screen. And let me share the next one. So the next one is basic concepts of enzymes. This is where we're starting to get into some more of the more complicated material in this area. So basic concepts of enzymes. I do want to just quickly go through the notes just to show you some of these diagrams that talk to you about changes in activity, pH, reactions that are occurring, the different types of reactions. This is one table that I think is very, very important for you to know. More so just this left-hand side and this middle one, oxoreductase, transferase, hydrolase, and so on. It's giving you an example of what it is doing. You're going to hear these types of names, these types of reactions throughout all of metabolism. The example is less critical. We'll get to these as we go through, but it is important to know that when you see something like a dehydrogenase, D is removal of a hydrogen, that's part of redox, part of oxidation reduction. It helps you to understand the reaction that is occurring. It is impossible for us to go through metabolism without having these long names, without having these processes, without having the chemical interactions. So what I would do my utmost is to try and help clarify and simplify these interactions. So don't get fixated on the name. What you'll find though is that those names are really quite descriptive. So lactate dehydrogenase is telling you what the substrate is, it's telling you the activity of the enzyme, it's telling you what it is doing, the type of reaction that it is doing. So it will actually help you to understand the process. This is a really nice diagram telling you in terms of delta G and what things are occurring, whether something will occur spontaneously. So if you're starting with something that wants to go to a product, it's going to be very easy because it's got energy already built in. Whereas if you want to go from something that's more complicated that requires energy input, it takes effort. Energy can be heat or it can be ATP as two of the classic examples. And then we talk about the rate. And then remember, there are different ways to express what is occurring in terms of product being generated from substrate over time in the presence or absence of an enzyme. So you can see here, you don't have the enzyme, A, you may not get the product at all. It may occur at such a slow rate that it's not viable for a cell, and you might get offshoots, you might get other products that are generated, which is why we use enzymes to accelerate. You have, there's only hand side, and there's no question when you have. So please keep asking. Okay, so stop. Okay, it seems like everyone is happy, which is good. Okay, are there any other questions? 8.2, you know that it has a net negative or net basic character because it's only migrated to that location. So it gives you additional information. So as I said, follows 1D, you separate them based on pH and is used to confirm purity of that particular mixture. So that's really quite an important way of determining purity of that particular protein and that construct. Okay. Are there any other questions? So feel free to unmute or put it into the chat relating to this particular lecture. Or just let me know on the screen with an emoji or something how you're feeling about this lecture. Thanks, Amy. Again, sorry, I'm probably mispronouncing your name. Everyone else? Myra, Chelsea, thank you. Okay, it seems like everyone's happy, which is good. And remember, these tutorials, if you've got content from last week's tutorial, please don't think, oh, I can't ask a question about last week. You can ask questions about the content. It takes effort. Energy can be heat or it can be ATP as two of the classic examples. And then we talk about the rate. And then remember, there are different ways to express what is occurring in terms of product being generated from substrate over time in the presence or absence of an enzyme. So you can see here, if you don't have the enzyme, A, you may not get the product at all. It it may occur at such a slow rate that it's not viable for a cell, and you might get offshoots, you might get other products that are generated, which is why we use enzymes to accelerate, be specific, and to then have a control point for that production. And this is what it's really the key point here. This energy hump, this is where the enzyme comes into play. It is reducing that transition state. It is making the amount of energy for that reaction to proceed. It is lowering that energy requirement, which is why heat might be enough, body heat might be enough for the reaction to occur, or a little bit of energy input in ATP or phosphorylation. If this wasn't here, you can see the energy state is much higher. And this transition state is that intermediate between substrate and product. It's where the two are bound, or the substrate is bound to the enzyme. It's the process of being acted upon, but it's not yet a product. It's still the substrate. It's in that middle sort of awkward phase, if you want to put it in that way. Okay, and then we talk about the enzymes site, and you can see that the enzyme, or the active site, very small part of the enzyme. There are multiple amino acids that come together that are the catalytic amino acids that allow the reaction to occur, which is all those reactions we saw in the previous table, but they're not linear. They might be throughout the protein, and they don't have to be linear because proteins folded, twisted, and come together, and that was really the basis of the last lecture, or the last tutorial. Different models of how they interact, so lock and key and induced fit, and it's probably somewhere between the two. We don't really know which is the way that it interacts, but we know that there has to be specificity between the two. And that also shows you the directionality of this diagram, that if you turn this any degree, apart from the shape, it just doesn't fit. And this is where that lock and key model comes into play. Regulation, because you need to be able to turn enzymes on and off, and how that can occur. So they're really the main points, and we will use that throughout the semester, because we're going to focus on enzymes. Everything that we move on, if I had to summarize biochemistry, it would be with the word enzyme. Enzymes are the key point for biochemistry, so you need to understand how it's occurring. So we're going to start layering and building on those ideas. So let me go to you. What's the relationship between an enzyme catalyzed reaction and the transition state of a reaction, which is that diagram that we saw before? Chat is open, and I can still see you on this screen, so please yell out. It might be glucose. Okay, here's an example. Glucose. Glucose is quite stable. One of the ways that we metabolize glucose is that it's acted on by an enzyme called glucose 6-phosphatase, or glucose kinase, I should say. You have to put a phosphate onto that molecule. So glucose, the end result is glucose with a phosphate attached. The transition state is where it's most unstable, because that reaction or that process is occurring. And by reducing that instability, it's more likely to proceed. That's where the enzyme, the kinase, comes into play. Helps to make it easier and for that to occur.
Hey Maurizio.
Yes.
I have a question. I just want to clarify about the transition state.
Yeah.
So because you know how the reactions are dependent on the substrates colliding in a specific way, from what I understand, so then they can make a formally bonds. Is that it? And then when they're binding to the active site, does that mean the catalyst is sort of positioning them in a way that makes sure they do the reaction in a more favorable way? Or
so it's this is a good example. I think you're on the right track. If you imagine this is the enzyme, this pocket here is the active site, and here is the substrate. You can see that, right? And so this is a very now that's assuming there's one active site. Then yes, energy that's required, less energy is required, so we're going to speed up that rate of the reaction. And if we don't have that, then yes, it may occur, but it might occur once every 3,000 years. And there's some examples in your textbook where key biological reactions that occur within the cell, if it didn't occur, the cell wouldn't be able to live, because it would take so long for that process to occur. It has to be enzyme driven. So Nathan, enzymes catalyze the reaction by stabilizing the transition state and in turn lower the activation energy needed for the reaction to occur. Perfect. Yep, that's really good. Thank you. That's a good answer as well. Okay, so I think that's really answering the next one. How do enzymes facilitate that formation of that transition state? It's that active site that we've spoken about. And as we just heard, location of the substrate where it's converted into the product, and it's usually a very, very small part of the total enzyme. And so with that diagram that I showed you in your notes that were coloured, and you've got 4 or 5 different amino acids, they're the key amino acids because when they come together and the protein folds into a particular shape, they're brought into a particular location. That location is what then determines whether the substrate is able to come in. And similar to the question we just had from Ira about the substrate binding, we said, oh, it's got this particular shape and the enzyme has the other shape that is able to interact. What makes that shape in the enzyme? If you imagine my fingers being the active site, each one of my fingers is an amino acid, and it just happens to be in a particular location so that the substrate is able to bind. If we mutate, right, that amino acid might go. So we've got now different interactions. Different substrates could potentially fit in, different substrates could be acted upon, or your preferred substrate no longer fits because it's no longer stabilized. This one, that thumb one, might be a really critical amino acid to stop the substrate from being dislodged and keeping it in there so that the reaction can occur. So by mutating it, it comes in but doesn't bind, the reaction doesn't occur, and obviously that's going to have a negative consequence on the cell. This last point that I wanted to talk about, water is excluded unless it's a reactant. This is really, really important. Our cells are obviously in an aqueous environment. All of the molecules are in an aqueous environment. But water can actually interfere with a reaction. And because that pocket is so small and the substrate can come in, it's actually tight enough interaction that any water that is there gets squeezed out. And so by doing so, it allows the reaction to occur. Now, where I've said unless it's a reactant, is that if you go back to that previous table, you'll see that one class of reactions is called a hydrolysis reaction. And this is where water comes in and is used to break bonds. Okay, so that tells you that that water molecule is part of that instability, and by having that water molecule there, it's able to break bonds apart, and that reaction might be where you've got 2 glucose molecules that are joined together. Having that hydrolysis reaction, and then you have those individual glucose molecules, for example, or fructose and glucose, whatever it is. 2 different groupings that are then broken through that reaction. And this last point is other amino acids serve as scaffolding. If I go back to my idea of the active complex, and we said that the fingertips of my hand are the active site, what am I referring to here? What's the other amino acids? It's the rest of my finger, it's the rest of my hand. If I didn't have those amino acids there, those fingertips wouldn't exist, they would just be floating. So those other amino acids provide the rest of that shape and that construct so that the substrate is then able to come in and be acted on. That's the idea. Okay, so how is the substrate bound to the active site? We've already seen this. You've got this particular configuration, a particular shape. So that's an important point. Only one shape is going to lead to productive reactions. Why? Because if you have a different shape, it may not fit. If you have a similar shape but it's missing key components, then they're not going to interact. So for example here, imagine you had a substrate, the natural substrate comes in, it's acted upon. You have another substrate that looks very similar, but it's lacking, say, a hydroxyl group. The amino acid that comes in is missing a hydroxyl group. That hydroxyl group might be the point of attack for the reaction. So even though it comes in, the phosphate that might be attached to that hydroxyl group can't occur, so nothing happens. What happens to the enzyme? Nothing really, except it's now delayed in its activity. So if it's able to do 100 reactions per second, if 50 times per second it's binding to a substrate that is missing that hydroxyl group, it's being unproductive. No product is being generated. So what am I talking about here? I'm really referring to an inhibitor. That other product that is coming in is actually inhibiting the activity of that enzyme because it's coming in, the enzyme's trying to do something, but it's not able to do its normal activity, and so it's being slowed down. We're actually decreasing or inhibiting its activity. So that's why that interaction is really important. And in terms of those interactions, we're talking about weak interactions, multiple weak interactions. Substrate comes in, it binds, and it is held weakly through multiple contact points. Once the reaction occurs, you want it to be able to be released. If those interactions were really, really strong, then once it came in, how are you going to get the substrate out? And it's no longer a substrate, it's going to be a product after the reaction has occurred. It will be stuck. And if you do that, you have now deactivated that enzyme. And again, that's a perfect example of how we can inhibit an enzyme. We can make a drug that when it comes in, it binds really, really tightly, and it now kills off the activity of that enzyme. So they're 2 different examples of why these processes have to be specific, and they also have to be weak enough for things to come in and out. So you can see ionic hydrogen bonds in particular are very weak, but you have multiple ones of them because there's lots of oxygen, hydrogen, and nitrogen atoms in amino acids. So lots of these, but they're very easy to make and break throughout the process. Okay, sorry, I've sort of covered a few there, but we had a bit of that content before, but this one I think will be a new question. In an enzymatic reaction in a test tube, so this is the key point, in a test tube, reaction will eventually reach equilibrium. And this is where you've got a steady state between perfect can inhibit an enzyme. We can make a drug that when it comes in, it binds really, really tightly, and it now kills off the activity of that enzyme. So there are two different examples of why these processes have to be specific, and they also have to be weak enough for things to come in and out. So you can see ionic hydrogen bonds in particular are very weak, but you have multiple ones of them because there's lots of oxygen, hydrogen, and nitrogen atoms in amino acids. So lots of these, but they're very easy to make and break throughout the process. Okay, sorry, I've sort of covered a few there, but we had a bit of that content before, but this one I think will be a new question. In an enzymatic reaction in a test tube, so this is the key point, in a test tube, reaction will eventually reach equilibrium, and this is where you've got a steady state between substrate and product. Why does this not happen in a living organism? What do you think? This is probably a bit of a harder question, just to make you think about what's occurring, and you need to bring in some of your first year chemistry into answering this type of question. Anyone want to have a go? Doesn't matter if you're wrong, it's tutorial. This is where we can discuss.
So I didn't go back to first year chemistry, but
Okay.
I said that if it's in a test tube, the reaction can occur without some of the regulatory mechanisms that occur inside a living organism. So I said the allosteric control as an example, that regulatory molecules that bind to allosteric sites that change the shape of the enzyme, thus altering the active site, inhibiting substrates from being able to bind to the active site. So yeah, it just won't get some of those mechanisms if it's in a
Okay.
in that.
No, that's good. Yeah, I think that's good. That's something that's occurring in a test tube. So Charlotte, negative feedback. I suppose that's very similar in terms of molecules that are being produced can provide a negative feedback. Yeah, Kirsty, product is used up in the body. Remember, this is in a test tube, so the question is test tube, so it's not going to be the case. So your answer is not on the right track simply because if we go to the answer, remember the level of substrate is not fixed within the cell, but it is within the test tube. So within the test tube, you might have, let's say it's 100 millimolar of substrate. The enzyme acts on it. As it starts to act on the enzyme, the level of substrate declines. What happens to the level of product? Goes up. So as you make substrate gets converted into a product. So as we heard, you could have allosteric control that the product comes back and turns off the enzyme. If we ignore that concept, right, what you'd see within the cell, that as substrate gets depleted, typically the cell is going to add more unless we have control mechanisms. So the cell wants to have a homeostatic level. So as it's produced, if we need 50 millimolar of product, once we get to 50 millimolar, we then come back and we inhibit. Now within the test tube, again, you're going to have a balance. Do you remember Lee Chantelier's principle from first year chemistry? Maybe not, but that principle is basically that if you have a substrate or a reaction starting, it's going to want to drive it through to the end. But when the two are in a balance, that driving force is going to be lost because the equation becomes more balanced. So as more substrate is produced or provided, more product is produced and diverted. So as a result, you start to draw more into the reaction, so you start to get this balance. And that's what would occur within a living organism. It cannot occur within the cell or within a test tube. What you've got is consumed. When you get to that balance point, you then reach that equilibrium. There's no more drive to go forward because you've got an equal amount of both. And so this is that process. as we heard with the explanation with the allosteric control, the product can turn off the enzyme. Because if you don't, the enzyme might continue to catalyze because there might be ample amounts of substrate. It will convert a lot of that substrate into a product, but if the cell only requires 50 millimolar, but it makes 500 millimolar, so 10 times as much, A, it's wasteful, it's a use of energy, and it's a waste of the substrate being converted into the product. But actually having too much product can be detrimental to the cell, because the cell sort of is in a state where it's got all of this molecule. Those molecules are, I don't want to use the word thinking or realizing, but if you think about it in that particular way, are reacting in a particular context, they may go off into other reactions, and you can have side reactions. And those side reactions are typically unwanted. They may generate a product that is not expected within the cell. So you want to try and keep things within the homeostatic level, because that's the optimal level. And as we've started to see with enzymes and enzyme kinetics, enzymes have a preferred level of substrate concentration. They work at a preferred rate of activity. Those requirements are within thermodynamic and also within equilibrium sort of constructs, and within that equilibrium range, what you would want at that equilibrium state. If you're too low, it sort of helps to speed things up, but once you get to a certain point, you want to be able to turn things off. So as I said, that's probably more of an advanced question, more of a deeper thinking question, but I think you're all on the right track. Even Kirsty, with your comment about being used up in the body, is perfect, still correct. The only difference was that the question was about the test you got within the cell. Perfect, that was spot on, so that's fantastic. Okay, I think, yeah, that was the last one for there. Is there anything, this one is a little bit more of a complex lecture, is there anything else in this lecture that you had as a question? Please feel free to, in the chat or verbal.
How do enzymes facilitate the formation of the transition state?
Yep.
I kind of went and talked about the classes of enzyme reactions. In a test situation, if that was to happen, would be lose marks entirely.
Do you want the good news or the bad news? Yes, it's probably not right. It's probably a little bit, because the question, if your answer was, oh look, here's an example, you've got a isomerase, you've got a dehydrogenase, you've got a hydrolase, and they all work because they bring the substrate in, this active site, and you sort of then use it as an example, then you've still answered it completely. If you just say, look, there are these different, there's redox reactions, there's dehydrogenase, there's ligase, there's hydrolase types of reactions, and that's all where you left it, then probably haven't answered the key point of the question, no. All right? But remember, no one ever passes or fails just on one question. And that's the whole point of these tutorials. Make your mistakes now. That's perfectly fine. I want to try and clarify and make sure it's all key. So does this answer that now make sense to you?
Yeah.
Okay, perfect. And thanks for verbalizing that too. Anyone else? Are we happy to move on to the last one? Okay, thank you. Now the last one. Just share screen. Here we go. This last one is now getting again into probably a bit more of a more complex topic. And I do want to go and give you a product. That's fine. That's fairly sort of linear and very simple. In some situations, that protein is made. What's that telling you? That it requires a diver to occur, although they're very rare. Is a reaction again. All right? Right. The bonds. Okay. And where they're important, I've highlighted those key points. Here, the coenzyme A, this is important for lipids. We'll talk about that in the following lecture as well. Okay. Now, I did want to dedicate that amount of time to talking to this because I think it is important. These are complex ideas and you've got to see them over and over again. So I really did want to position it so you've got an idea of why we're seeing these molecules, what are they doing? And also to stop you from being nervous or anxious or scared of all of the chemistry that is going to come through. The chemistry is there to illustrate ideas, to illustrate principles. So please don't think, oh, we want to remember all of this chemistry. No. And even when we get to enzyme names, there might be some that I want you to know. The vast majority, we're not going to ask you to remember. Okay, so hopefully that reduces some of that tension. Now, just quickly, because I've just noticed the time, apoenzyme, holoenzyme, remember, apoenzyme, inactive enzyme, no cofactor, no substrate. Typically, we're not talking about that one. We're going to be talking about the holoenzyme. So it's active, it's got the cofactor, it's ready to go, but there's no substrate. So that could be hemoglobin. It's got the heme group, it's got the iron atom, it's ready to go, but there's no oxygen. You breathe in, again. So I really did want to position it so you've got an idea of why we're seeing these molecules, what are they doing? And also to stop you from being nervous or anxious or scared of all of the chemistry that is going to come through. The chemistry is there to illustrate ideas, to illustrate principles. So please don't think, oh, he wants us to remember all of this chemistry. No. And even when we get to enzyme names, there might be some that I want you to know. The vast majority, we're not going to ask you to remember. Okay, so hopefully that reduces some of that tension. Now, just quickly, because I've just noticed the time, apoenzyme, holoenzyme, remember, apoenzyme, inactive enzyme, no cofactor, no substrate. Typically, we're not talking about that one. We're going to be talking about the holoenzyme. So it's active, it's got the cofactor, it's ready to go, but there's no substrate. So that could be hemoglobin. It's got the heme group, it's got the iron atom, it's ready to go, but there's no oxygen. You breathe in, oxygen comes in, it binds, and now able to mediate its activity, and off it goes. So as I said here, this is the one that we'll use unless we talk about it. But just thinking through the course, this is the one that 99% of the time we're going to talk about. Bring this in, because you do see it, You might see it in your lecture notes or your textbook in particular. It's not really our focus. Okay, back to you as a group. Discuss the importance of metal ions in enzymatic reactions. Give examples of metalloenzymes and metal-activated enzymes highlighting their role. So we'll just take off the first one. That tells you the importance. A third, require metal ions for activity. What else would you say in this context? If this part is confusing and you don't know where to start, please also let me know. Charlotte, metal ions are cofactors that facilitate redox. Yep, that's good. So within the enzyme, good. What else? So some examples. The most classic ones are going to be, as you see there, iron, manganese. I actually don't even have magnesium, but magnesium is one of the key ones. If you think magnesium, you think DNA polymerase as one of those enzymes that has that critical factor. And so there you go, metal activated enzyme. So more loosely bound, and you've got magnesium. So the iron in the two different states is within the hemoglobin, where it's much more tightly bound, and then you can have more loosely bound types of interactions as well. Okay, so let's keep going. So metal ions bind to the sub iron, manganese. I actually don't even have magnesium, but magnesium is one of the key ones. If you think magnesium, you think DNA polymerase as one of those enzymes that has that critical factor. And so there you go, metal activated enzymes. Or loosely bound, and you've got magnesium. So the iron in the two different states is within the hemoglobin, where it's much more tightly bound, and then you can have more loosely bound types of interactions as well. Okay, so let's keep going. So metal ions bind to the substrate, regulate the conformational change to orient them for a reaction. Similar to the question that we had earlier from Ira about the substrate coming in, the metal ion helps in that process. Rather than just sort of spinning, if it's able to orient and say, well, you need to come in this orientation to work, it makes that a much more efficient process rather than just waiting, oh, let's wait until you get to a configuration that fits. Oh, here we go, now we're binding. It helps to direct that process. As we heard from Charlotte, mediates redox reactions which are at the core of biochemistry. So remember, oxidation, reduction. It stabilizes or shields negatively charged groups, and that's important. And may unite formation of metallocoenzymes. Perfect. So the heme group, right? So that's that formation of those intermediates. You've got those different groupings. And those two enzymes, or two examples, are the ones that I gave you earlier. Catalase. This takes hydrogen peroxide, H2O2, breaks it down into water and oxygen molecules. This enzyme is actually present in bacteria. And what bacteria do is that they use this to break down hydrogen peroxide, which is used by your immune system to kill the bacteria. So it's a way of it sort of evading. You make the catalase, the white blood cell is trying to kill the bacteria, and it's just being converted to water and oxygen, and the bacteria is just happy. Nothing happens to it. Here you've got hexokinase is working on a group substrate with 6 atoms, hexo, and it's a kinase. It adds phosphate onto those groups. And we'll talk about phosphorylation in depth throughout the semester. Okay, what about coenzymes and non-protein or organic molecules? What are we thinking here? I'll show you the first one. Activated carriers. When we see the term activated, it means that that molecule is ready to perform a function. It may have a group that has been added to it, like a phosphate group, and by adding that phosphate group to it, it makes it more likely to be reactive. Okay? Now I'm just going to speed up a little because I've noticed the time and I don't want to interfere with your next course, or your next teaching. So special carriers of electrons, that's certainly an activity. Accept from one and donate to the other. We saw that with NAD, we saw that with FAD, taking the 2 protons. So that was a good example. Important for oxidation, such as carbohydrates. Glucose is oxidized to give you ATP. And here are the ones that really, I would highlight these. These are the critical ones that you need to know because we're going to see them time. Time again throughout the semester. Okay, significance of coenzymes being regenerated. I touched on this before. Regeneration means that synthesis is not required or resynthesis is not required. If they were consumed, the cell would have to spend a huge amount of energy to make them. And because this is at the core of metabolism, all of your metabolic activity is just going to drop because you're spending 20% of your time just making these things that are being consumed, but they're core for business. So if you can recycle, it's going to be much more efficient for the cell. Much more efficient, but it also allows for other reactions to occur within the cell. Those cofactors that we see there, NAD+, NADH, etc., don't just have to be constrained to that process. They can go to a different part of the cell and undertake that type of reaction that might be in the mitochondria. It might be in a different organelle within the body or within the cell. So it gives you additional flexibility. Two exceptions, though, two examples are coenzyme A, and we'll see this when we come to talk about fatty acids. CoA is recycled. It's a derivative of acetyl-CoA. This is one that you will hear about a lot, and again, succinyl-CoA, but mostly acetyl-CoA. Acetyl-CoA is a key output of the citric acid cycle, the TCA cycle, which is the core of the metabolic pathway that leads to ATP synthesis. And here, it's actually a different pathway. But remember, these are exceptions. So these are the very rare occurrences that we're seeing. The other one is NADPH, and we will talk about this when we talk about the pentose phosphate pathway. It is the only other unique situation that we'll really focus on. Here, this has to be replenished through this pathway. It is regenerated rather than being recycled because there's limited pathways or limited opportunities for NADPH compared to NADH to be generated, so it's much more specific. And this last one here, examples of activated carriers, different pathway. But remember, these are exceptions. So these are the very rare occurrences that we're seeing. The other one is NADPH, and we will talk about this when we talk about the pentose phosphate pathway. The only other unique situation that we'll really focus on. Here, this has to be replenished through this pathway. It is regenerated rather than being recycled because there's limited pathways or limited opportunities for NADPH compared to NADH to be generated, so it's much more specific. And this last one here, examples of activated carriers, probably a redundant question because we've covered it, and that's why I said see previous diagrams, and that's why I went through, I'll just go back up, these diagrams. Here are the examples of the structures. Here are the, which are not critical, apart from what are they doing, but it's also telling you what they are, their basic activity in terms of biochemistry and metabolism, and their role within that process. So this one is a little bit more heavy. It is more biochemistry, it is more chemistry content, it is a bit more of a more complicated area, but what I've tried to do is just simplify why are we seeing, what is their main role, what is the importance of these molecules in basic cell metabolism, okay? Don't overcomplicate things because it's very easy, particularly in this context, to get very easily confused. Having said that, do you have any questions? I don't see anything in the chat. Answer to the fourth question. There you go, Naomi. Okay. Anyone else have a question? Just on 1103. So remember, always go back to your learning objectives on the front page of the lecture. Always go back to those basic concepts that I'm going to refer to. They are always going to be the basis of questions, always going to be the basis of my focus when I come to assess you. Make sure you understand those key points, and then we'll start to add those additional layers. If you don't understand those key things, it's going to make it very difficult for you as things are progressing. Okay, just double checking last opportunity. Naomi, thanks, Ira, no problems. I'll explain the fifth question. So this fifth question is really just going back to these previous diagrams, Naomi. So these ones, just to highlight, these were the ones that we were talking about. So it's a bit of a redundant question, just going back to these different equation. It's called the Michaelis-Menten model. And this describes the kinetics of lots of different enzymes, and it's the one that we'll talk about the most. So in this example here, or this idea, the moles of product generated per second, so remember this is the rate, so the amount of product generated in a given unit of time varies with substrate concentration. So remember, substrate plus enzyme gives rise to a product, and it can also go backwards. So the amount of product is dependent on the concentration of substrate, what you're starting with. But this is true when the amount of enzyme is kept constant, and so this is explicit. So we know that most biochemical reactions are going to be catalyzed by an enzyme, and enzymes convert a substrate. Now, this terminology, S within these brackets, this is the correct use of reference to substrate, so you'll have seen it in previous lectures where I've done that. So enzymes convert substrate into a product, which is a P, and the product may actually be an intermediate in a multi-step pathway. So if we imagine that A goes to B, that's your product, but then that goes on to give you another product, and so on. And you can have this cascade for as long as you want, or as long as is needed within the cell. So each one of these is a product, but then it continues to become the next product in the cascade as you go until you reach the endpoint. And the conversion of substrate to product is a time-related event. So what we do as biochemists is that we talk about the amount of substrate that is converted per unit of time, and that might be minutes, might be seconds, it might be larger or smaller than that, but typically we will talk about it in the context of minutes. So when we talk about enzyme activities, they're therefore understood in the terms of their rate of the reaction. So this is rate, remember, is activity per unit of time. So how much do we catalyze in a certain period of time? This whole area of study is called enzyme kinetics, and it is the study of enzyme catalyzed reactions, and it's an approach to understanding their function and space there. Now, in terms of understanding how enzymes work, there's a famous equation, and it's called the Michaelis-Menten model, and this describes the kinetics of lots of different enzymes, and it's the one that we'll talk about the most. So in this example here, or this idea, the moles of product generated per second, so remember this is the rate, so the amount of product generated in a given unit of time varies with substrate concentration. So remember, substrate plus enzyme gives rise to a product, and it can also go backwards. So the amount of product is dependent on the concentration of substrate, what you're starting with. But this is true when the amount of enzyme is kept constant, and so this is fixed. So there's a fixed amount of enzyme that's able to catalyze. So as we increase the amount of substrate, more will be available up to a point, and therefore you can vary the amount of product. So if we look at this diagram here, what we have on the horizontal axis is substrate concentration, and this might be, say, molar. And we've got the initial reaction, so looking at velocity, or the speed at which the enzyme is working. So as you can see, as we start to increase the level of enzyme, the level of substrate, it goes up and up and up very quickly, and then it begins to reach a plateau. Now, we see this dotted line, this is the maximum velocity, this is the maximum rate or velocity of the enzyme. But what you'll notice is that even if we keep going and add normal substrate, we actually never reach Vmax, or this maximum velocity. Because it can be a bit of a theoretical thing, in the sense that the enzyme can never operate at 100% efficiency because it's always going to be occupied in substrate. Other products or the byproduct of the assay can come back and interfere with enzyme activity. But this is the basic idea. We'll increase substrate as enzyme is fixed, and we'll get this change in reaction. So for an enzyme reaction, what we will see is this explanation. We've got our enzyme, and this is a very similar equation to what I've shown you before. Enzyme plus our substrate come together to form this enzyme-substrate transition state, and we've spoken about this in the previous lectures, the transition state. This is where, remember, this is where the energy has to come in. This is what determines whether the enzyme is able to mediate its activity. At this point, it's able to then move forward, and the enzyme gives rise to the product. Now, when we have K1, K2, these are rate constants. This is just the rate at which this first reaction takes place, and then the second one, and then the reverse is called K-1 and K-2. It's just the reverse rate constant, because we've got double-ended arrows, which means that we can go from enzyme substrate to enzyme product, but you can also go backwards. So, K1 is the rate constant of the enzyme substrate transition complex. So it's this initial starting point. K2 is the rate constant of the product. And as I mentioned before, the negative signs are just for the reverse reactions. Now, for most of what we're going to be talking about, we can probably ignore the negative reaction. We're going to have an enzyme, it's going to act on the substrate and give rise to a product, but it's got this intermediate step. So really, we're just interested in these two at the top. So if we measure the amount of product that's generated at varying levels, we see what we see here in figure 6.2. That as product goes up over time, at low substrate, so this is low substrate, this is high substrate, we have this initial reaction. So it goes up slowly, keep going, eventually you're going to reach a maximum amount. You can see that at substrate level 4, this is pretty much flattened out. These have yet to reach that, but they're getting there. We'll talk about V0 in a second. So, as expected, the amount of product increases over time, and you would think this because if enzyme is there, substrate is there, over time the enzyme is going to be able to take the substrate, convert it to the product, so therefore the amount of product will increase. But what we saw with that diagram, in all cases, there is a time point that is reached where we see no net change in substrate or product. So this is where we have the equilibrium or this plateau phase. So you can see here, we've really started to max out here, we've started to max out here as well, and it becomes very obvious now the really high substrate concentrations that are starting to flatten out. And this is that equilibrium between substrate and product. Now remember, one way that we can adjust this is that we can change either side, either the amount of substrate or the amount of product. Here, substrate has caused a limiting effect. So if we reduced product, so we took product away, then there would be more substrate and we would continue to have conversion. So that's an important idea to think about. Now, as I mentioned, for simplicity, we ignore the reverse reaction and we assume that the enzyme substrate complex, so that transition state, will lead to product being generated. Now, it is the relationship between enzyme activity and substrate that is described by the Michaelis-Menten equation, and here it is. So we saw V naught before, I've got these other parameters. I'll write them out even though it's down below because it's good to just have this clear. So V max is the maximum velocity, and Km is the substrate concentration equal to 50% of V max. So how much substrate do I need for the enzyme to be working at 50%? So V naught relates to the enzyme's initial reaction velocity, okay, and there are units. And so in this example, micromolar of product per minute. So this is the initial velocity. Vmax, as I said, is a theoretical maximum. So that's an important word, theoretical maximum velocity. So again, micromole of product per minute, so the amount of product per time. So in this case, these are still going to be rates. Substrate is the concentration of the substrate. And again, this is going to be whatever the unit is, but in this case, we can see that we're using micromolar. And Km is the substrate concentration, which, to be consistent, is micromolar, at which the enzyme is functioning at 50% of Vmax. So to what I've written, or exactly the same as what I said before. So what does this tell you? The equation describes the variation of enzyme activity as a function of substrate concentration. And you can see that here. Substrate divided by substrate plus Km, so this is going to be a larger number. This tells you the relationship between the two and how this then impacts on Vmax. So Km is 50% of Vmax, and this is one way that we can plot it. We have substrate concentration versus velocity, and you can see that the enzyme activity goes up, keeps going, begins to plateau. This is the theoretical value. And where we've got 50% of this curve, this is what is Km. So this could be, for example, 10 micromolar. But again, as I've written in the legend, but also here, just remember, these two are not touching. But the problem we've got is, we said Km is half of Vmax. If we're not calculating, or we can't actually determine what Vmax is, how do we then work out Km? Because these two are never going to meet, then this is going to be an approximation. So there's a way that we can fix that. Now, these two parameters, why do we have them? They're used to compare catalytic activity. And we do this to compare between enzymes. But what it also allows us to do is to identify changes in enzyme structure and function under different conditions. So we can put different molecules into an assay and then see, do they inhibit the assay, do they modulate? So modulate might mean that it actually increases the activity, whereas an inhibitor will decrease the activity. So by having some parameter, we can actually work out, does the amount of substrate that is required, does that change? So is Km being affected, or does the velocity change? Does Vmax change? And so these are the two parameters that we'll be looking at in the practical. Now, as I mentioned before, Km and Vmax, Km is derived as a function of Vmax, so whatever Vmax is, it's the amount of substrate that gives you 50%. But as I said, based on that graph above, if Vmax is never reached, well, we really can't be super specific with Km. So how can we overcome this? We can do this by taking the reciprocal of substrate and the velocity, so 1 over V, and plotting these values on x and y axes respectively. And this will give you a graph called a double reciprocal plot, or a Lineweaver-Burke plot for the two scientists. Now, what does a reciprocal mean? It's just 1 divided by S, or 1 divided by V. That's it. So 1 divided by this converts it, and then allows you to plot these two values. This gives you a Lineweaver-Burke plot, and this is the classic graph. So on the bottom, we have substrate, so it's 1 over S, Burke plot for the two scientists. Now, what does a reciprocal mean? It's just 1 divided by S, or 1 divided by V. That's it. So 1 divided by this converts it, and then allows you to plot these two values. This gives you a Lineweaver-Burke plot, and this is the classic graph. So on the bottom, we have substrate, so it's 1 over S, and this is the zero substrate. And at the vertical, we have 1 over V naught. So this is velocity. So this is velocity, and this here is referring to Km. Now you have your data points, and just ignore the negative for now, but you have some experimental data, so you have results that fall along, let's imagine they're perfect data points, like this. You then draw your line across and you hit the intercept. Now, at zero substrate, you're going to have zero activity, because obviously there's nothing to convert. But we can extend the line back to the negative space, and what we're looking for is this intercept here. The intercept is called -1 over Km. Now, don't get confused by this. This is quite simple. It is minus because we're in the negative axis, and we can actually ignore the negative because you can't have negative substrate. So it's 1 over Km, and we know this is Km relates to substrate concentration, so it's just the amount of substrate here. Similarly, this intercept on the vertical axis, this is 1 over Vmax. This is just telling us what is the velocity of this enzyme at multiple substrate points. Now, depending on the enzyme, and depending on what we've included with the enzyme, this parameter here and the Km will change. And those changes then tell us something about what is occurring to the enzyme and its activity. So just to repeat, -1 over Km, we ignore the negative because you can't have negative substrate, and this is Vmax. Now remember, if you have 1 over V or 1 over S, very large numbers are going to give you a small number. So 1 over 10 is going to be 0.1, but if you have 1 over 100, it's going to be 0.01. Okay, so it's going to be a bigger value. So these numbers here are reciprocal, so they're inverse of what we'll be talking about later. Now, the Km of an enzyme will depend on the substrate, but it also depends on the pH and the temperature. And these were two parameters that we saw in the previous lecture where we had the graph right at the beginning of the lecture showing how the two can relate, and also the ionic strength of reagents. Now, this is just an example, so some sample Km buffers of different enzymes, and as it says here, note the wide variation in Km. So chymotrypsin, this is the substrate. You don't need to know what the substrate is, but this is just what it works on. 5,000 micromolar. So this is 5 millimolar. Okay, so that's a lot. Lysozyme only needs 6 micromolar. Beta-galactosidase, 4 millimolar. Carbonic anhydrase, 8 millimolar. And penicillin, or penicillase, is 50 micromolar. So you can see quite a spectrum, or quite a difference. You can just compare 6 to 8,000. So each enzyme has different levels of substrate requirements. Now, let's think about this. When substrate levels are low, and they're low compared to the Km, so if we're saying that the Km is 8 millimolar, say for carbonic anhydrase, okay, and our substrate is at 1 millimolar, so we've got much, much less than 50%. What's going to happen? If you think about the enzyme, it's going to be very sensitive to changes in substrate, but it'll actually display very little activity. Why? Because remember, we need 8 millimolar for it to get 50%. So 1 millimolar, it may only have 5%. So if we double this, it may only go up to 10%. So it's only a little increase. It's only as we start to get closer and closer that we'll see more of an effect. Similarly, when the substrate is at Km, this is when we have optimal activity. So 8 millimolar, this is going to be the sweet spot, if we put it that way. And then the enzyme is going to be sensitive to changes. So even a fraction more is going to give you more activity. And when substrate is above or significantly above Km, activity is still going to be high, but it's going to be insensitive to changes in substrate because it's already operating at close to maximum. So activity is high, it's running as fast as it can, it's catalyzing as many reactions as possible, but you can change the amount of substrate, it doesn't matter. So just as if I was to say to you, run slowly, okay, and I give you a gentle push, that gentle push is going to make you speed up. If you're running as quickly as you can and I give you a gentle push, it's really not going to do a lot to what you're doing because you're already going at your maximum, you can't go any faster. So it's the same idea here with the enzyme. So what does this mean? What this means is that cells will have or operate an enzyme with a Km that will match the physiological level of substrate. So if an enzyme, whatever the enzyme is, doesn't matter, in the cell has a substrate, and that substrate level is typically at 0.1 molar, so 100 millimolar, its Km will be close to that. Because what that means is that this is the normal physiological level that you would have within the cell. This is when the enzyme is going to be operating most efficiently and therefore most reactive to the needs of the cell, and it's not going to require huge amounts of additional substrate. So this is quite an important idea. What about Vmax? Vmax tells us the turnover number of an enzyme. How many reactions can it generate per unit of time? So let's say per second. So number of substrate molecules converted into product per unit of time. So remember, this is another way of just talking about rate. And this is when the enzyme is fully saturated with a substrate, so it's operating at its maximum activity. And we can calculate this. So if we've got 10 to the minus 6, so this is 1 micromolar carbonic anhydrase, it forms 0.6 molar, so 600 millimolar of bicarbonate per second. Put a line there. When it is saturated, so it's maximum. So what is Vmax? Well, it's going to be the amount, so the amount of time over the amount of enzyme, so we divide. Those are going to be cancelled out. So you see 0.6 molar at 10 to the minus 6 molar of enzyme, you've got 600,000 reactions per second. So this is a reaction every 1.7 microseconds. You can see it's phenomenally fast, but you don't need a huge amount of either enzyme or actually substrate. Okay, if we just go on to this table here, you can see turnover number. We just saw carbonic anhydrase. This is quite a very fast enzyme. You can see that there are others that are also as fast. But also, let's look down here, lysozyme, 0.5. So it takes 2 seconds to catalyze a single reaction. You've got quite a spectrum of different reactions. Notice down here, DNA polymerase. This is the enzyme that copies DNA. Only 15 reactions per second. So quite slow. But remember, there's proofreading. There are other issues around copying DNA, so you don't want it to go significantly different. So the key point here is that there are massive differences between individual enzymes and their reactivity. Okay, a lot of content there. Make sure you go through it. Make sure it's clear. I'll stop there because we'll move on to talk about reactions of a single enzyme as well as multiple enzymes. So, when we think about reaction, what we will see is that there are typically multiple substrates. So what I've said to you before is that we've got a substrate plus an enzyme. This will give us the enzyme substrate intermediate, and then you end up with enzyme plus product. That's the normal reaction. And what we've got here is one substrate, one enzyme, giving us to a product, and we sort of stop there, we don't go any further. But as it says here, most reactions actually have multiple substrates, so there's going to be multiple of these. And remember, this substrate converts into a product. This product can now become a substrate for the next reaction, and so on. So what we spoke about with the enzyme kinetics relates to an enzyme binding to a single substrate and generating a single product. But as we see here, most cases, 2 substrates come together to yield 2 products. Now, when we come to think of this approach, how can this occur? These reactions can either be sequential, so one occurs, then the next one, or both occur at the same time. This is called a double displacement reaction. It's important to understand this because we're going to see this when we get to metabolism in future lectures. So here is our enzyme. Here is NADH. Remember we spoke about that as a cofactor in the cofactor lecture. And here is pyruvate. So the enzyme with NADH and pyruvate goes forward, converts it to lactate. So pyruvate is converted to lactate, and the NADH goes to NAD+. So this is a sequential. You've got one series of reactions going together, but you can see that you've got one molecule, 2 molecules that are required. In this example here, this is a double displacement reaction. So here we've got spartate and oxaloacetate. These 2 come together to give rise to a reaction. You've then got this intermediate, which is this group in here. This in turn interacts with another group, so alpha-ketoglutarate, and that in turn continues on to give rise to the final product, which is glutamate. So we've got double things happening essentially at the same time. These 2 coming together, then another series of reactions. So as it says here, catalytic reaction occurring, forming 2 products, and the enzyme and then the individual products are released. That release point is important because it is the binding to the enzyme which allows for these reactions to take place. So in the double displacement reaction, which is what we've got here, the first substrate is aspartate, and we've seen that here. It binds to the enzyme, and the first catalytic reaction generates a product, oxaloacetate. So substrate, and now this is our product. So that now disappears, as we see here. Now, leaves and the second substrate, alpha-ketoglutarate, so here's another second substrate, binds to the enzyme and a second reaction takes place. So here we've got that reaction taking place. These two come together to generate our glutamate, and that is then released. So you can see you've got multiple reactions taking place. Individually, they are the same. The only difference is the order and the manner in which they are occurring. So in a sequential reaction, all substrates bind to the enzyme before the product is released. We saw that with the first example, but with the double displacement, we saw that the first product was released, and then additional substrates are binding to the enzyme before being released. And that was the oxaloacetate. That was released, and then we had the alpha-ketoglutarate coming in. Okay, again, that's a reasonably short one, but I'll stop there because we're now going into another concept that I want to put into the next recording. So see you in the next one. So we've spoken about allosteric enzymes, and we now need to apply this to enzyme kinetics again. So most enzymes will follow Michaelis-Menten kinetics. If substrate is present, product is going to be formed. So if the enzyme is there, substrate will get converted to product. However, activities of enzymes also have to be regulated. If this reaction here, we know it's going to occur, but if it just kept going and going, what would end up happening is that this level would be unregulated, the level of substrate would decline, and this would cause problems to the cell, because if the cell is sensitive to substrate and it drops, that's problematic. If we have too much product, at first it's actually wasteful, but also this can cause other issues within the cell, and we'll see that in a moment. So we need a way to regulate metabolic activity, and one way to do this is just to regulate enzyme activity. And we know this because enzymes control the flux of metabolites, the flow of metabolites. And we saw that in the previous diagram where we had the different substrates moving through. So you can see that if the enzyme is turned off, it's going to affect the flow of all of those products through the pathway. And so through these pathways, we've got allosteric enzymes. And remember, an allosteric enzyme are regulated by the products of the pathway. So this product can come back and regulate the enzyme. And by doing so, it can either turn it on or turn it off. And this way, we've got a self-regulatory loop. We've got a way for the amount of product to come back and turn off the enzyme. As the level of product declines, the inhibition drops and more substrate gets converted. It's a very nice balancing act between the two. Now, if we look at this diagram here, 6.7, this is showing us 5 enzymes in a pathway. It doesn't matter what they are, just you've got enzymes and E1, E2, etc. What we can see and what we need to understand is this term, first of all, the first committed step. The first committed step is the first reaction within a pathway. It is the one that will initiate or propagate the entire pathway, and it is typically the point where we want to control the pathway. Because if we stop the first enzyme, then the remainder of the pathway can't begin, can't continue. And the way that I like to think of this is a bit like dominoes. So if you imagine you've got your 5 dominoes, we push the first one, it knocks these over. But if we put a block here, and we push this one over, it goes against the block, nothing happens. So this is the most critical component. None of these can operate on their own because they're dependent on this very first one. So that's the basic idea. So first committed step is A going to B, which is what we see here. The final product in this example, down here, notice the blue line, it goes back and it has shown as a negative sign. So this is acting as an inhibitor. The final product of this pathway comes back and inhibits the first committed step enzyme. And by doing so, we can turn this off. If we turn this off, then the activity of the subsequent enzymes are affected because the amount of substrate is going to decline. As the level of the final product declines, this inhibition is removed, enzyme activity returns, and we have reactivation of the entire pathway. So a really clever way of controlling the whole process. That's one approach. Now, if we look at 6.8, so here we've got 2 pathways coming together to generate a final product. So here, the final product that we're interested in is K. All right, doesn't matter what it is, that's just our final product. So in this case, product K can inhibit both enzyme 1 and enzyme 10. So let's have a look. Here is our enzymes, we're going along, it starts, generate this product. This product can then go into another pathway, so it feeds into G, H, and so on, which gives you J and K. So where are we inhibiting? 1, which is here, and 10, which is here. First point, notice that F comes back, or sorry, F goes on to this pathway. As it comes through, K comes back and blocks the very first enzyme. So again, this is showing that negative loop. F can also then participate in a different pathway. So that's what this line is showing you here. I'll draw it again, just showing you coming into a second pathway. This again is the first enzyme in this second pathway. So again, we want to control it. But notice here that product F is a positive. So what this does is that it stimulates. So when F becomes high, it stimulates the activity of this enzyme and we get more product being generated. They come together to go to K. Now, as we saw, if we followed K, it comes back, it inhibits. We've also seen that if we go down this way, K can inhibit the starting point of this enzyme as well. And even within the pathway, we've got this molecule, so I, another endpoint of this cascade, it can come back and actually stimulate this enzyme. So notice one side can inhibit, one side can stimulate. This is important because if we've got this product, so product F stimulating here, I goes up, we want this to keep feeding, so we've got this amplification. And similarly, when there's too much, we want the ability to inhibit, and this is where K is able to do so. This is a little bit more of a complex sort of an explanation, but all that we've done is we've introduced another pathway here. And the second idea is that rather than always inhibiting, we can also stimulate a pathway, and that's what we see here with the plus and the minus. So what is the take-home message? The take-home message is that typically the endpoint of a pathway, so here's the endpoint of this pathway as it feeds into another, endpoint here of this pathway, or the combined pathway, can usually go back and somehow modulate the previous steps. And again, this is all just showing us allosteric control. So an important control point for this complicated mix of enzymes. Now, as we've got down here, products F and I stimulate the activity of E1 and E10 to limit wasteful production of other intermediates. So that's the other benefit of this approach. By stimulating, you don't have the build-up of other intermediates. It's then wasteful. They can also become toxic to the cell. Now, when we look at allosteric enzymes, they don't actually follow Michaelis-Menten genetics. And this is because they're regulated by products that are generated via the pathway, as we saw. They can either be negatively regulated or they can be positively regulated. So how can we explain this? There's a separate model. It's called the concerted model. And this aims to explain the regulation. So if we just look at the blue line, as we increase substrate and we've got reaction, you can see that reaction substrate goes up very, very quickly, this enzyme, and then it plateaus very quickly. So normal Michaelis-Menten. This enzyme is very sensitive to substrate, so we don't need a lot for it to have massive activity and then just plateau. But notice with the allosteric enzyme, we need more substrate and there's more of a tapered effect, because as product is being generated, it's going to start to inhibit the activation. So this gets delayed. We need more and more substrate before we can get more and more product, and then we again reach this endpoint. So it's a slightly different shape of the curve. Okay, I'm going to stop there because the next part is a little bit more complicated, and product is being, as we increase substrate and we've got reaction, you can see that reaction substrate goes up very, very quickly, this enzyme, and then it plateaus very quickly. So normal Michaelis-Menten. This enzyme is very sensitive to substrate, so we don't need a lot for it to have massive activity and then just plateau. But notice with the allosteric enzyme, we need more substrate and there's more of a tapered effect, because as product is being generated, it's going to start to inhibit the activation. So this gets delayed. We need more and more substrate before we can get more and more product, and then we again reach this endpoint. So it's a slightly different shape of the curve. Okay, I'm going to stop there because the next part is a little bit more complicated, and we need to start thinking about how we can explain those reactions in terms of Welcome back. Now, what we do know is that typically allosteric enzymes have multiple active sites on different polypeptide chains. So multiple active sites means that you can have multiple binding of substrate. Okay? The way that this is explained is that an enzyme exists in two different configurations or two different states. And the term that is used within biochemistry is called the R state, which is relaxed, and the active form of the enzyme. So when it's relaxed, it's active, and the other is the T or the tense state, and this is when the enzyme is inactive. So R equals active, T equals inactive. Okay, so keep that in mind. Now, when we don't have substrate, which is probably going to be rare, but if we imagine that there's insufficient or just no substrate, these two are going to be in equilibrium because there's no binding. So half will be active, even though there's nothing to catalyze. So occasionally, the tense state will turn into the relaxed state due to energy, which might just be heat that's present in the environment. Because remember, there's part of that activation energy that exists within the body, but also a requirement for the enzyme. So the model requires that all active sites of the enzyme must be in the same state. They have to be either tense or relaxed. You can't have a hybrid. And then lastly, substrate binds more readily because the enzyme is now available and able to bind the substrate. It binds to the R form, which remember was the active form, and less so to the T form. So what this is telling us is that when it is in the active site or active form, it's able to bind the substrate. When it's in the T state, it's inactive, it doesn't bind the substrate, and it can exist in these two different configurations. So let's have a look at the diagram. Here we see an enzyme with 4 active sites on 4 subunits. So that's all that is being shown here. So this is the enzyme in the T form, and this is the enzyme in the R form. So you can see in this example in part A, most of it's in the inactive form, because it's in the squares. And we've got one that's in the active state, but there's no substrate. We can see here, enzyme with 1 substrate bound. We can see that in this example here, this example there. And notice we start to see more of the active components. We now have a situation that we've got 2 now being bound, more and more, but now we've got more with 1, now with 2, and as we keep going, we've got 3, and then ultimately all of them are going to be activated and have substrate being bound. So what does this mean? So if we look here, most enzymes are in the T or inactive state. So that's at the starting point. So little substrate will bind to the R form because there's not much of the active form present. So what we're trying to do here is explain why doesn't low substrate give rise to a lot of activity. It doesn't, because there's really not a lot of active enzyme or enzyme that's willing or able to bind to substrate. That's what we saw here in A. As we start to increase substrate concentration, it will bind to the active form. And you can see that here and here. As more sites move into the active state, it becomes easier for the substrate to bind to the active state. And you can see here, we now start to have more and more active state and more being bound to at least one of the active sites. And then as more enzyme is in the active form, the binding of substrate becomes easier and we have more of this interaction. It's a bit of an interesting way of trying to explain what's going on. But again, think of it from a very much a broad view. We're trying to just say, how does substrate bind? Why does low levels of substrate not cause activation? Because what we've got is an enzyme that's in a state where it is not receptive to the substrate. But as we start to increase the amount of substrate, more of the binding takes place and more of the activity is seen. Now, if we look at this diagram here, allosteric enzymes display threshold effects. So what we can see here is substrate, millimolar, and velocity as a fraction of total Vmax. So the blue line is a Michaelis-Menten enzyme. So you can see here, as we increase substrate, it goes up and up and up, and then tapers off, just what we've seen previously. And we look here, it's at 2 millimolar. So the higher levels of substrate, 27% fold, are required for a change in Vmax compared to an allosteric enzyme, which is in red. Now, if we look at red, you can see that you go down and you don't need a lot for some activity to be seen. So you've got some activity here, it then shoots up as a fraction of the total activity, 0.8. So what we're saying here is that we've got substrate at 0.8 divided by 0.1, the 27-fold difference. And here, it's just a fold-fold, so it's not exactly those numbers, but a 4-fold difference between the two. So, how do we explain the ability of these enzymes to respond to changes in the environment? So if we go back to figure 6.7, we had the product, the regulatory product F. It alters the equilibrium between the tense and relaxed states. As something that stimulates the assay, or stimulates the reaction, binds to the R form, it stabilizes the active form. It increases the interaction between the active form and the substrate. If this occurs, more likely for the substrate to be acted upon to generate a product. What about an inhibitor? Well, the inhibitor is probably going to do the opposite, isn't it? So the inhibitor binds to the tense form. So remember, this is the off state, or inactive. And by stabilizing the off state, it decreases the chance of the active form binding to substrate. And so we can see this in this diagram here. Here's an example. So effective regulators on the enzyme aspartate transcarbamylase. So here is the enzyme, so the rate of carbamyl aspartate being formed, and this is the substrate, aspartate. So ATP is an allosteric activator of the enzyme, and it stabilizes it in the R state, which is the blue line. So you can see that in the presence of 2 millimolar ATP, at lower levels we start to get stimulation and we get more product being generated. CTP, on the other hand, is an inhibitor. So cytidine triphosphate stabilizes the T state, and you can see here that we get a depression. So even higher levels, we're producing less product because we're inhibiting, so making it harder to bind to its substrate. And the black line is in the absence of both of these. But notice, 2 millimolar ATP stimulates a little bit, but only 0.4 inhibits quite a lot. So we've got this interplay between stimulation and activation taking place. Okay, bit of a complicated lecture, but as always, we'll go through it in the tutorial, so make sure you come with your questions. You've got your summary here of the lecture, and we've got this is now lecture 7, and we're going to talk about inhibitors of enzyme activity. And so really the idea is how enzymes are inhibited by drugs, things that we would take from a therapeutic perspective. So we need to talk again about the properties and structures of enzymes. We'll talk about how enzymes enhance biochemical reactions, so some coverage of material that we've covered before. We'll talk about the induced fit model, and know that enzymes are catalyzing an enormous number of diverse reactions within the cell. And for this to take place, each reaction requires the formation of that enzyme substrate transition state. So remember, substrate plus enzyme initially gives you enzyme substrate complex, and that ultimately gives rise to the product. So they all have to go through here. This is a way that we can inhibit by keeping it at this particular point. Now, in terms of enzymes and inhibitors, the catalytic activity of an enzyme can be inhibited by metal ions or small molecules. It doesn't need to be something large. It can be a very small disruption to the three-dimensional shape or to the active site that can interfere with the activity or the catalytic activity of an enzyme. These inhibitors can be endogenous, so things that are produced by an organism, or exogenous substances such as drugs. And these can all act as inhibitors and as a regulatory control mechanism for the pathway. Now, depending on what we're talking about, there are different classifications on how the inhibitor functions, and so you're going to have to understand reversible or irreversible inhibition. And quite a simple idea in terms of what can occur, but we'll go into it in a bit more detail. And then irreversible, reversible inhibitors, what we see are associated with a rapid dissociation from that enzyme inhibitor complex. And we'll talk about the 3 different types that are seen. But what you'll also see in the explanation from that enzyme inhibitor complex. And we'll talk about the 3 different types that are seen. But what you'll also see in the explanation below is some material that we've seen again, so the enzyme fit model, but also the kinetics and also the Lineweaver-Burke plot, the changes. So we're going to start to bring those previous lectures together to really have a nice idea of how they all fit together. So first one is competitive inhibition. And what we have here, this is where we've got, say, a substrate plus an enzyme, gives us an enzyme, substrate, intermediate, and a product. If we've got another molecule, let's just call it X, that is similar to S, then it may also form this complex. And by forming this intermediate complex, so it would be the X, rather than generating the product, the amount of product that is generated is going to be reduced. So this is how a competitive inhibitor. The inhibitor X competes with the normal substrate to bind to the enzyme, and by doing so, it lowers the amount of product. So this type of reaction is characterized by a rapid dissociation of the enzyme inhibitor complex. So the enzyme can bind the substrate, or it can bind the inhibitor, but it can't both be bound at the same time because it's only got one active site. So if it's only got one spot for binding, then it's really a bit of a lottery between the two. Now, it's only a lottery when the concentrations are low. If we have a variation in the concentration, so we've got an excess of inhibitor compared to substrate, then more likely we're going to be binding the inhibitor as opposed to the substrate. So as I mentioned before, the competitive inhibitor resembles the substrate and binds to the active site. Now, what does resemble mean? It means it resembles it based on its structure or its chemistry. It's able to bind to the active site. The net result is that the rate of catalysis decreases because more and more enzymes that bind to the substrate are reduced, and more enzymes that bind to the inhibitor are increased. So we've got more binding to the inhibitor and less binding to the substrate. Can we overcome this? And the answer is yes. Inhibition can be relieved by increasing the amount of substrate. So if we imagine we've got 10 molecules each of substrate and inhibitor. Here we do have a 50-50 chance. We might pick this molecule, we might pick this molecule. If on the other hand, we keep the 10 molecules, but we've now got 1,000 molecules of inhibitor, you can see that this is going to be much more likely to be bound by the enzyme than this. So this is where the inhibition occurs. If we now say, okay, well, we're going to change that, we're going to have 1 million molecules of substrate, then that is now far greater than the inhibitor. So what we can do is we can relieve the inhibition because it just happens that there's more chance it's going to bind to the substrate than it will to the inhibitor. So the relative concentrations of these is really important for what can occur. So here's the idea. We've seen this before. This is the induced fit model. Here is our enzyme. Here's the substrate that is bound into the active site. And here is a control point on the side, but we'll see that later. It's not important for today or for now. You can see here is our competitive inhibitor. It's also binding to the active site. There's no difference in the shape of the enzyme, but notice that the inhibitor only partially resembles the substrate. It's enough to bind, but what that means is that this inhibitor blocks the binding of the substrate and it can no longer enter. So what does this look like when we look at the different data, the Michaelis-Menten data? So now you can see I'm starting to bring in the previous results. So we increase substrate and we look at rate. So S versus V, similar to what we've seen before. If we look at the black line, no inhibitor goes up and up and up. We reach our maximum and we're there at that time. If we now have, let's say that these are micromolar, so one micromolar of inhibitor, you can see that there's a reduction. 5 micromolar, a greater reduction. 10 micromolar, even further. So you can see that there's a dose-dependent effect. So just as we said up here, we can relieve the inhibition by increasing the amount of substrate. We can make the inhibition greater by increasing the amount of inhibitor, and that's what's shown here. 1 micromolar, 5 micromolar, 10 micromolar. We get more and more, and we can keep going. Okay, we went say to 50, for example. Now, how does this affect the Michaelis-Menten and the 1 over V plot? So if we look here, no inhibitor present. This is our normal line of the fit. This is where our Vmax comes in. This is our minus 1 over Km normally. With our competitive inhibitor, you notice that the slope of the line has changed. So what this means is that we're still intercepting at Vmax, so velocity is unchanged. What does that mean? It means that the rate of conversion of substrate to product remains the same, because the enzyme is still acting at the same speed. It's just that in one reaction it might have substrate, in another reaction it might have inhibitor. But notice, by changing the slope, we now change the Km. The Km is getting smaller, it's getting closer to here, to 0. Now remember, this is an inverse, so a smaller number here means a higher level of substrate. So 1 over a small number equals a large substrate concentration. So 1 over 0.1 is going to equal a substrate of 10. So you can see small number, as that number gets smaller, the amount of substrate is going to get bigger. So as this gets more and more vertical, you're going to need more and more substrate for this to occur. This explains the type of inhibition that is seen. You need to understand the inhibition that's shown on the left, but as well as the one on the right, because this is going to be important in our enzyme kinetics practical. But also, this is the basis of treatment, of therapies. Okay, when someone gets sick and is given a drug and it interferes with an enzymatic process, we're somewhere within these examples that we're going to be covering today. So you need to understand how these all interact and how they work. Okay, now I want to give you an example here. What we have at the top, this is methotrexate. This is a competitive inhibitor of the enzyme dihydrofolate reductase. And we will talk about this in the nucleotide metabolism lecture at the end of the semester. Now, this is, as it shows down the bottom, structurally similar to the natural products, dihydrofolate here on the left and tetrahydrofolate here on the right. So if we look at it, you can see that this ring structure is very similar to this ring structure here. The remainder is very similar. And we've got the same issue on this side. These are all very similar, but are slightly different in this context. Where are the differences? Here, here, and here. They're the only differences. But those differences are sufficient that they're able to bind to the active site but prevent the formation of the natural substrate or the natural product. Okay, I'll stop there. We'll come back on non-competitive inhibition in the next recording. Do so at different sites, and it must be at a different site. Hey, welcome back. So we just spoke about competitive, but now let's talk about non-competitive inhibition. So you can tell by the name this is going to be different and probably going to work by a different mechanism. So in this case, the inhibitor and the substrate can both bind simultaneously to the enzyme, but they do so at different sites, and it must be at a different site because we just said above that you can't have the inhibitor and the substrate binding to the same site. So just as if I wanted to sit in your seat when you're sitting there, that's physically not possible. So it's the same idea, there must be at different sites. So the inhibitor, by binding, or decreases the turnover rate of the enzyme. Now, because the effect is not at the active site, by changing the amount of substrate, it doesn't do anything, it's at a distant site. So if we look at the same type of diagram, you can see we had our substrate binding to the same point, but remember we spoke about this point here, this is that control point. This is where the non-competitive inhibitor binds. By binding here, notice that there is a shape change. And what was the term that we used? This is also a conformational change. Shape change or conformational change. You can see it's quite different. And by doing so, you can see that the substrate's not binding as effectively, the shape of the enzyme is different, and it's quite distinct. So how does this affect the parameters that we've seen before? So, substrate versus velocity. So no inhibitor, as before, goes up, exactly the same shape. At 1 micromolar, you see we get an initial effect, and then it plateaus, and then again, 5 and 10 plateaus, but you can see it pretty much blocks it immediately. So there's not much of an increase, even at a moderate concentration. If you look at the 1 over V, you can see the no inhibitor, we've got the same idea, we've got Vmax here, Km. Notice that again, the slope has increased, but we're now at the same Km, because remember, substrate does not change. So you expect that Km would be the same. The only thing that changes is the velocity, so therefore, it is intercepting at a higher point. So remember, higher point, bigger number, means lower velocity, because it's an inverse, it's the opposite. So we can see that we've now got a decrease in the rate of the reaction for that particular enzyme. So the previous one and this one here are two examples that you need to know, because this result, or this data, explains the chemistry or the biochemistry of the enzyme. Now, the one that is being shown here, uncompetitive, this is quite a rare or less common type of inhibition, and to be honest, I was in two minds whether to give this to you, but it is one that is starting to become a bit more apparent through drug therapy, so the reason for its inclusion. So uncompetitive is also substrate dependent, but what we see here is that the inhibitor only binds to the enzyme substrate complex. So it only binds to enzyme substrate complex as opposed to binding in place of the substrate. So the binding site for the inhibitor only appears or becomes present when the enzyme binds to the substrate. And because of this, we can't overcome it by increasing the substrate concentration. So again, standard diagram, here is our enzyme active site, substrate is bound. Here we've got substrate binding. That binding causes a small change at a local level, and the uncompetitive inhibitor is now able to bind. Now notice if we had the uncompetitive inhibitor with an empty enzyme, so if you imagine this was gone, there's no binding. So it only occurs when substrate binds. Now again, let's have a look at the profile, so substrate versus velocity. You can see that the no inhibitor is the same shape, and in the presence of the non-competitive, this looks very much like uncompetitive, this looks very much like a non-competitive inhibition. But when we look at the 1 over V versus 1 over S, you can see that both parameters have changed. Km is shifted, and correspondingly, so has Vmax. So quite an interesting, this is a relatively rare approach. You won't see this in any of the practicals. This is quite a unique sort of situation, but it's an important one to understand because of the mechanism of action. It's quite an interesting way that it's able to do this. Now, if we talk about irreversible inhibitors, what about these? How do they differ? These differ to what we've seen before because these bind tightly. Remember before, we were talking about binding, but then also release. So short-term binding because we've got the interaction of either enzyme or substrate and inhibitor. So here we've got bind tightly to the enzyme, and this is non-covalently and only rarely disassociates. So once bound, it remains attached. So it binds covalently with a functional group or to a functional group that is essential for an enzyme's activity. So remember, the functional group are the amino acids in the active site. So by binding to those amino acids, we're blocking the activity. So two examples, penicillin, this covalently modifies the enzyme transpeptidase, and by doing so, it prevents bacterial wall synthesis. And the other one is aspirin. Here we've got again covalent modification, this time of an enzyme called cyclooxygenase, and this reduces the synthesis of inflammatory mediators. In both of these, how do we get resolution or back to normal? We need to make the protein, and remember by that I mean the enzyme, new again. So what does that mean? It must be re-synthesized. Okay, once it's bound, it is basically destroyed. So it's only when that protein is removed, the inhibitor is removed, that we then continue the process. And that also goes on to explain why with these two examples we have repeated administration. And that's true for most drugs. Okay? In this case, we have repeated administration because once these enzymes are cleared and new ones are made, we need to have the inhibitor there to inhibit them. With the other examples, substrate levels may change, the enzyme, the inhibitor may decline, and therefore the inhibition is also going to be lost. So by having that repeated administration, we continue